We use cord-derived MSCs. So we have developed our own potency assays. So we look at the cell's functionality and how they interact with different immune cells and through which pathways they are signaling. So we do an overall assessment of the cells. They should not be active through a single pathway. They should have the ability to censor the environment and to increase the level of that specific pathway that is driving the disease. So the rationale behind ProTrans is to make an overall assessment of the cell's capability and pick the cells and donors that meet our high set criteria. We also have a pooled product, so we use cells from 5 donors to get a bigger batch. All in all, this makes our product very reproducible, and we know that it's very potent because we test it all through the production. And we get very large batches since we produce cells from multiple donors. So the advantages are specifically that we can make it very repeatable, because we don't want to exhaust these cells. We don't culture them too long. We only culture them for 3 passages, and then we get new material. So this is not a traditional way of just growing cells over and over again, because we see in our potency assays that we can grow them from many more passages, but they will lose their potency, and that we can measure in the potency assays that we use to select the donors. So one of my challenges is to persuade the efficacy of these cells. We have shown that now in data, but since early time MSCs that were produced from bone marrow, they were produced for a very long time. Those cells, I'm sure, lost their potency down the line. So we have produced the next generation of MSCs using the selection algorithm and also starting over every time for every batch to keep the cells very primitive and naive.